Thank you, everyone. You may have your seat again. Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu, Namo Buddhaya, Salam Kebajikan, Rahayu. I would like to say good morning and a very warm welcome to the visiting professor event with the theme of Advancing Mushroom Science from Biology Cultivation to its Bioactive Application. This visiting professor event is a conference that proudly presented to you by the Faculty of Science and Technology at Universitas Terbuka, especially Biology Study Program on this day, 19th August 2026. First of all, allow me to extend our warmest greetings to the honorable speakers, Professor Norihiro Shimomura, and also Professor Tsuyoshi Ichiyanagi. So our speakers, Professor Shimomura and also Professor Ichiyanagi, coming from Faculty of Agriculture, Tottori University, Japan. And also I would like to welcome our honorable guest today, the Dean of Faculty of Science and Technology at Universitas Terbuka, which represented by the Vice Dean for Academic Affairs, Dr. Sita Alifarihati, MSE, the Head of Edible Mushroom Biotechnology and Nutraceuticals Research Group from National Research and Innovation Agency, Dr. Rini Rifani, PhD, the Vice Dean for Finance, Resources, and also General Affairs at the Faculty of Science and Technology at Universitas Terbuka, Dr. Anda Inggit Winarni, MSI. Vice Dean for Learning Services, Student Affairs, and also Partnership at the Faculty of Science and Technology at Universitas Terbuka, Dr. Pepi Rospina Pertiwi, MSI. Head of Biology Study Program, Dr. Elizabeth Novi Kosumaningrum, SSI, MSI. Both of our moderators of this visiting professor event, Dr. Iwan Saskiawan from National Research and Innovation Agency, and also Ms. Hikmah Zikriani, MSI from Universitas Terbuka. And I would like to say to give a warm welcome to all participants who joining us physically here in Auditorium of Faculty of Science and Technology at Universitas Terbuka, and also all of the participants who joining us virtually through Zoom and also YouTube. And I would like to introduce my own self. My name is Safira. It is such an honor for me to guide this visiting professor event. So, ladies and gentlemen, to further know about the Faculty of Science and Technology of Universitas Terbuka, we would like to watch the profile video of the Faculty of Science and Technology of Universitas Terbuka.
Universitas Terbuka merupakan perguruan tinggi negeri ke-45 di Indonesia yang juga merupakan pelopor dan inovator pendidikan jarak jauh di Indonesia. Dalam upaya untuk terus meningkatkan pendidikan jarak jauh yang berkualitas dunia, layanan Universitas Terbuka didukung oleh 39 kantor daerah yang tersebar di seluruh Indonesia dan 1 pusat pengelolaan luar negeri. Fakultas Sains dan Teknologi Universitas Terbuka merupakan fakultas dalam bidang sains dan teknologi yang bertujuan menghasilkan lulusan yang kompeten, mandiri, dan kompetitif di bidang sains dan teknologi. Awalnya, fakultas ini dinamakan Fakultas Matematika dan Ilmu Pengetahuan Alam. Kemudian pada tahun 2019, berdasarkan Permenristek Dikti, berubah penamaan menjadi Fakultas Sains dan Teknologi atau FST.
Selamat datang di Fakultas Sains dan Teknologi Universitas Terbuka. Universitas Terbuka atau disebut UT menyelenggarakan proses pembelajaran secara jarak jauh dengan menggunakan berbagai media teknologi informasi dan komunikasi. UT sebagai institusi pendidikan tinggi berkomitmen dalam menyediakan akses pendidikan yang fleksibel, terjangkau, berkualitas, dan berdaya saing luas tanpa batas bagi seluruh masyarakat Indonesia. UT akan terus berupaya untuk meningkatkan mutu dan daya saing di kancah nasional maupun global. Fakultas Sains dan Teknologi atau disebut FST merupakan salah satu fakultas di UT yang dapat dipilih sebagai jembatan untuk mencapai karir tertinggi, profesional, dan pribadi yang berkarakter, sehingga dapat berkontribusi terhadap kemajuan bangsa Indonesia. Perkembangan mahasiswa FST yang meningkat dari tahun ke tahun menunjukkan kualitas dan kepercayaan masyarakat terhadap FST UT yang tumbuh positif. FST menawarkan 8 program studi jenjang sarjana di bidang sains dan teknologi yang meliputi program studi S1 Biologi.
Program studi S1 Biologi mempersiapkan lulusan dengan penguasaan mendalam terhadap prinsip-prinsip biologi untuk mengoptimalkan potensi keanekaragaman sumber daya hayati Indonesia dengan menghargai kearifan lokal dengan didukung literasi digital, kemandirian, dan etika profesional yang kuat.
Program studi S1 Matematika.
Program studi S1 Matematika menghasilkan pembelajar mandiri yang dapat menerapkan konsep dan prinsip matematika dalam menyelesaikan berbagai masalah matematika dan terapannya. Lulusan program studi S1 Matematika FST UT banyak berkiprah sebagai praktisi di bidang pemerintahan, industri, dan organisasi kemasyarakatan, asisten peneliti maupun akademisi.
Program studi S1 Statistika.
Program studi S1 Statistika dirancang untuk menghasilkan lulusan yang memiliki kompetensi akademik di bidang statistika dan penerapannya, serta mampu berpikir kritis, analitis, dan sistematis, sehingga dapat berkiprah di sektor industri, pemerintahan, maupun penelitian.
Program studi S1 Perencanaan Wilayah dan Kota.
Program studi S1 Perencanaan Wilayah dan Kota atau PWK memberikan pengetahuan dan keterampilan dalam menyelesaikan berbagai permasalahan sosial, ekonomi, lingkungan, dan infrastruktur dalam suatu kota dan wilayah. Program studi PWK UT membersamai mahasiswa belajar untuk dapat menjadi seorang perencana yang handal, yang siap bekerja di berbagai bidang termasuk pemerintahan, perusahaan, maupun penelitian.
Program studi S1 Sistem Informasi.
Program studi S1 Sistem Informasi dirancang untuk menghasilkan lulusan yang memiliki pola berpikir analitik dan sistematik di bidang sistem informasi yang dapat diterapkan untuk memberikan solusi terhadap berbagai masalah dalam masyarakat.
Program studi S1 Agribisnis.
Program studi S1 Agribisnis menghasilkan lulusan yang berdaya saing unggul yang berwawasan pengembang masyarakat di bidang agribisnis. Program studi agribisnis mencetak lulusan yang memiliki kompetensi sebagai wirausaha, manajer, dan pengembang masyarakat agribisnis.
Program studi S1 Teknologi Pangan.
Program studi S1 Teknologi Pangan menghasilkan lulusan yang kompeten di bidang industri pangan berbasis sumber daya lokal dan memiliki wawasan kewirausahaan. Lulusan prodi teknologi pangan dapat berkiprah sebagai manajer, wirausaha, regulatori, praktisi di bidang QA, QC, PPIC, R&D, dan bidang lainnya di industri pangan.
Program studi S1 Sains Data.
Program studi S1 Sains Data dirancang untuk menjawab tantangan zaman dan mencetak para ahli data handal di era revolusi industri 4.0. Para lulusan dibekali keterampilan ilmu statistika, pemrograman, visualisasi data, machine learning, dan big data analysis agar siap bekerja sebagai data scientist untuk menyelesaikan masalah real di berbagai sektor seperti bisnis, industri, dan sosial.
Desain kurikulum yang dinamis pada setiap program studi selalu di-upgrade dengan menyesuaikan perkembangan iptek dan dinamika pasar sehingga dapat memenuhi kebutuhan tenaga kerja profesional di Indonesia.
Fakultas Sains dan Teknologi Universitas Terbuka atau FST UT memberikan berbagai layanan akademik yang mendukung kesuksesan belajar jarak jauh, meliputi layanan SSO My UT, Virtual Reality, Lab Praktikum, Perpustakaan Digital. Mahasiswa dapat mengasah keilmuan dalam bidang sains dan teknologi melalui berbagai kegiatan, antara lain visiting lecturer, webinar nasional, seminar nasional Saintek, seminar internasional. Selain layanan akademik, Fakultas Sains dan Teknologi UT juga mendukung kegiatan kemahasiswa untuk mengasah soft skill dan hard skill mahasiswa melalui program kreativitas mahasiswa, program pembinaan mahasiswa wirausaha, program Merdeka Belajar Kampus Merdeka, Olimpiade, Dispor Seni, Inkubator Virtual Sains dan Teknologi, prestasi mahasiswa. Dalam mewujudkan pendidikan jarak jauh yang berkelas dunia, FST UT telah menjalin kerjasama dengan dunia kerja serta universitas ternama di dalam dan luar negeri. Kerjasama dilakukan dalam bentuk pengajaran, penelitian, dan pengabdian bersama, serta magang mahasiswa.
Belajar di UT memberi saya fleksibilitas untuk kuliah sambil bekerja, sekaligus melatih disiplin dan pola pikir kritis. Yang paling penting, UT mengajarkan keterbatasan, bukan halangan untuk bermimpi besar. Buat kamu yang sedang kuliah di UT, percaya diri, terus belajar, dan jangan takut melangkah lebih jauh.
Berkuliah di Fakultas Sains dan Teknologi Universitas Terbuka merupakan pengalaman yang berharga bagi saya. Sistem belajar yang fleksibel memungkinkan saya untuk mengatur jadwal kuliah saya kapanpun, dimanapun sesuai dengan kebutuhan. FST UT bukan sekedar tempat kuliah, tapi tempat membangun masa depan.
Sekali lagi, selamat bergabung di FST UT untuk masa depan yang lebih cerah. Faculty of Science and Technology, Kreatif, inovatif, kolaboratif. FST UT.
Give round of applause to the Faculty of Science and Technology at Universitas Terbuka. So, ladies and gentlemen, before moving on through the next agenda of today's event, let us begin our honorable event today by praying, seeking blessings for the activities that we will embark upon today. Please start praying. Thank you. May God give His blessings and grant our wishes. So, ladies and gentlemen, let us hear a welcome or opening remarks that will be delivered by the Dean of Faculty of Science and Technology at Universitas Terbuka, which represented by the Vice Dean for Academic Affairs at the Faculty of Science and Technology at Universitas Terbuka. So, ladies and gentlemen, please welcome Dr. Sita Alif Farihati, MSI.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Alhamdulillahirrahmanirrahim. Here I will deliver the remarks of behalf of the Dean. So first, Alhamdulillah, on this very fine day and in this good health, we can gather in this forum, and it is also our privilege that we can join in today's visiting professor. Our esteemed of speakers, Professor Norihiro.
Recording in progress.
PhD, and Professor Dr. Engineering Tsuyoshi Ichiyanagi. Good morning. From Faculty of Agriculture, Tottori, University, Japan. Our Honorable Dr. Iwan Saskiawan from BRIN as first moderator, and Mrs. Rina Riviani, PhD. Thank you very much for your welcome. Our Honorable all Vice Deans in Faculty of Science and Technology, FST Universitas, Mrs. Inggit and Mrs. Pepi Rospina. All the head of study programs in the Faculty of Science and Technology, Universitas Terbuka, especially Dr. Elizabeth Novi Kosmaneningrum, SSC MSc, as head of biology study program. And then our second moderator, Ms. Hikmah Zikriani, MSc, and all lecturer, students, and all participants who are present in this visiting professor to attend on-site and online. Firstly, I will praise to Allah Subhanahu wa Ta'ala for His grace and blessing, allowing us to gather in this important forum to discuss featuring a highly engaging and relevant topic, advancing mushroom science from biology, cultivation to its bioactive application. Okay, and then here I will talk about Universitas Terbuka. Yes, Universitas Terbuka possesses distinct characteristic as an open and distant education with a vast student reach spanning various regions across Indonesia. So this creates immense opportunity to develop research and innovation routes in Indonesia biodiversity and local potential, including the potential offered by various types of fungi. Okay, we must ensure that biodiversity does not merely remain an object of research and scientific publication, it should be transformed into innovation, technology, product, and solution that generate added value for society. Therefore, I hope today's event will spark a world of new ideas. So I hope today's we can remark the beginning of stronger institutional and academics partnership between Universitas Terbuka and Tottori University. So we can explore various forms of collaboration, including joint research, joint publication, or exchange of faculty members. Then maybe we can join international seminar, yes, and we can develop of cultivation, technology, and fungal utilization and others. So I believe is the true meaning that international academic collaboration is not merely about exchanging knowledge, but about jointly creating new knowledge and generating a greater impact. Finally, I would like to thank all the organizer and participants for organizing this event, and I hope this event smoothly and benefitly us all. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Sita. Please remain on the stage because everyone who attend this event physically here in auditorium, we will have a photo session together before moving to the main agenda of today's event. So I would like to invite to all participants to come to the front, please. Dengan ini saya mengundang kepada seluruh hadirin untuk dapat maju ke depan karena kita akan melakukan sesi foto bersama terlebih dahulu. So once again, I would like to invite to all participants who joining us here physically in auditorium, please come to the front because we will have a photo session together. Okay. Boleh dipercepat Bapak Ibu, kita akan melakukan sesi foto bersama, mohon berkenan. Okay, once again, with honor, I would like to invite to all participants, all lecturers, the students, and also participants who are joining us here physically in auditorium to have a photo session together. We can have 2 rows or 3 rows for the men. You can have your position at the front. Mungkin untuk yang bapak-bapak atau mas-mas boleh berjongkok di depan. Oke. Oke. Apakah masih muat? Boleh dibantu diposisikan. I would like to ask the official to help us to guiding this photo session to make a memorable photos together in this beautiful occasion for today's event, the visiting professor. Okay, and I would like to inform that we will have 3 photo sessions, one with the formal pose, second with the semangat pose, and the third is salam UT. You with the right hand and T with the left hand. Like this. Okay, and I would like to ask to all online participants to open your camera because not only participants who joining us here physically, but we will have a photo session together with all participants who joining us virtually through Zoom. Okay, if you may to all participants that joining us virtually through Zoom to open your camera. Okay, once again we will have a 3 pose for this photo session. The first is formal pose, the second is semangat pose, and the third is the iconic pose of Universitas Terbuka Salam UT. Okay, yeah. Okay, so we will now continue to the first photo session with the formal pose. I would like to help you to count down from 3, 2, 1. Okay, and the second pose is the semangat pose. Okay. I would like to count down from 3, 2, 1. Okay, and the last pose is the iconic pose of Universitas Terbuka with the UT logo by using your hand. Okay. Okay, we will count down from 3, 2, 1. Okay, let's give round of applause to us all here. So, ladies and gentlemen who attending this event physically here, you may have your seat again. Okay, so before moving to the main agenda of today's event, the presentation sessions, so I would like to inform to all participants who joining us physically here or virtually through Zoom or YouTube, we will have two presentation sessions by having two honorable speakers that already joining us physically here in auditorium, and each presentation sessions will be followed by Q&A session. So, to all participants who joining us here physically, if you want to ask questions to our speakers, you can raise your hand and the committee or the officials will pass you the mic and you can ask directly to our speakers. And to all participants who joining us virtually through Zoom, you can write down your questions through the chat box, or you can ask directly by clicking the raise hand button at the Zoom. So, ladies and gentlemen, we will now proceed to our first visiting professor session entitled The Development of Mushroom Science in Japan. And I would like to ask to all participants who joining us virtually through Zoom, please keep your microphones in muted mode during the presentation session. And once again, I would like to remind, if you have any questions, please submit them through the Q&A session by using Zoom chat box, or you can use the raise hand button if you want to ask directly. So the first presentation will be presented by our first speaker, Professor Norihiro Shimomura, and this session will be guided by Dr. Iwan Saskiawan from National Research and Innovation Agency. So, ladies and gentlemen, please join me in welcoming Dr. Iwan Saskiawan.
Okay, thank you, Master of Ceremony in this event. Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. Good morning, ladies and gentlemen. Today, the first presenter in this program is Professor Norihiro Shimomura from Tottori University. First, I will read the CV of him. He graduated from Faculty of Agriculture, Tottori University in 1992, and from 2013, he became the professor in Fungus Mushroom Resources and Research Center, Faculty of Agriculture, Tottori University, and in 2019 until 2023, he became a Dean of Faculty of Agriculture, Tottori University, and from 2023, he became the principal of Tottori University affiliated junior high school in Japan. Okay, for the Professor Norihiro Shimomura, I would like to ask you to come in the front. Dozo, Sensei. Yeah, here. Okay, Professor Shimomura, your time is about 45 minutes presentation and 15 minutes for the discussion. No, it is your time, Professor.
Good morning, ladies and gentlemen. I'm Norihiro Shimomura, Tottori, Japan. So thank you very much for a very, very kind introduction for Dr. Iwan. So firstly, I would like to say all over the participants, this floor and also the internet class. Firstly, I would like to thank to the organizing committee of Terbuka University, especially for the Dr. Norma Sensei, Dean of this area. So I also want to say thank you very much for the great opportunity to me, Dr. Yuan, and to introduce this Terbuka University as a visiting professor. So I would like to, firstly, I would like to start my presentation. So today the topic is, you can find in the slide, development of mushroom science Japan. My topic is basically the basic information, not specific information. So please move to the next slide, please. So my topic today is especially for the four topics. One is the trend of mushroom in Japan. Next is a case study of shiitake mushroom. And third topic is a case study of maitake mushroom in Japan. And finally, when we have enough time, I would like to inform the cultivation strategy of ectomycorrhizal mushroom sorrel. It's a main work of my study. So when we would like to have some topics within the 45 minutes. So firstly, I would like to we move back this one. So this is this fourth topic. But when we have enough time, we can join. I would like to inform the cultivation strategy. Next slide, please. First, I would like to inform that the mushroom cultivation was led by the private company, not the government. So you can find some company in this slide. You can find the spawn company. We have many spawn companies, shiitake, especially for the kinko shiitake, mori, hokken, kinox, akiyama, fujishuki. They developed the shiitake, especially for the shiitake breeding, only providing a spawn only. And so when the farmer purchase the spawn from this spawn company, they cultivate the mushroom. Another area is a mushroom company. Maybe you know that Hokuto Mushroom Company, a big company in Japan. And this is a bunashimeji mushroom. Bunashimeji and king oyster mushroom. This company make a substrate and make a spawn and make a sawdust medium and provide a culture bag and cultivate mushroom and sale by the company. All of the mushroom-related work were carried out by only one company, Hokuto Company, Ikiguni Company, and Ichimasa Kamaboko Company. And Nakano City, some agricultural association. So we have two types of the mushroom society. One is a spawn company, while another one was mushroom generated company. Next slide, please. So this slide, you can find this slide in some recent mushroom production in Japan, you can find the most abundant mushroom were found in the enokitake mushroom, winter mushroom in supermarket, and bunashimeji mushroom, and shiitake mushroom, and maitake, and king oyster, and enoki, and dry shiitake. The most abundant winter mushroom. But shiitake is, to tell the truth, shiitake is the most big mushroom in Japan because shiitake is a third, fresh shiitake is a third. But we can find in the dry shiitake in the bottom side, this shiitake, fresh shiitake and dry shiitake. Dry shiitake is we can count it by 10 by. Because of dried material. When the dry shiitake originated from the more and more big fresh mushroom. So the cultivation, main mushroom is the shiitake in Japan. So next slide, please. So you can find this recent trend of the shiitake mushroom. You can find in the I would like to say that the green line, it means wood log cultivation, log cultivation. At 1985, mushroom were cultivated by using wood log at that time, but this wood log you can find significantly decrease. And oppositely, you can find the purple line. It's a sawdust medium, sawdust bag culture shiitake. So this method was developed in the 1993 or 1995. So this mushroom significantly increased after the developing of the shiitake mushroom. At the same time, in the developing for the shiitake by sawdust medium, cultivated sawdust increased. In the same time, the imported mushroom you can find in the red line. Red line is imported from the China. So when the mushroom, shiitake mushroom developed in Japan, the same at that time, China also produces many mushrooms. But this China mushroom were increased only until 2000, but recently decreased in Japan. But now in the recently, the production of the shiitake is basically sawdust-based mushroom. And wood log mushroom were decreased, and imported mushroom is also decreased. This is the trend of shiitake in Japan. Next slide, please. So this slide shows the table of the mushroom, fresh mushroom cultivation, and this slide indicates that how the sawdust-based shiitake cultivation increased with time. So the sawdust, 1987 is only 5% of shiitake were cultivated sawdust, but 2009 about 80% of shiitake were cultivated wood sawdust cultivation. Next slide, please. And this slide shows the cultivation, shiitake cultivation and imported shiitake. In Japan, we have basically 3 types of shiitake. One is the wood log. It's a traditional mushroom in Japan. Another one is the sawdust-based cultivation. Left side, bottom left side. And right side bottoms, we can find China. They called bag log bug logs cultivation. Bug log, I can confuse bug to logs. Why the bug log matched the but they saw that later they produce bug culture, but resemble to log. So they call the bug log cultivation. So you can find in the left top table domestic and imported mushroom. And the fresh mushroom were basically recently the fresh mushroom were sawdust. But important point is that dry mushroom, domestic dry mushroom Japan is basically cultivated only wood log, not sawdust based mushroom did not use for the dry shiitake. But imported mushroom were sawdust based cultivated one. So the imported one is basically used for the powder slice. But dry shiitake, all mushroom were as used as a gift for year end, middle year end, the gift. So next slide, please. So I would like to emphasize to the fruiting process to understand the basic information for the mushroom cultivation. This slide a very, very important slide to understand the mushroom cultivation process. This process is basically firstly developed by the wood log cultivation, but this process is easily applied for sawdust cultivation. You can find first left mycelial learning stage. It's a mycelial propagation stage. After the mycelial propagation, mycelial learning, and then some fully mycelial colonization, the mycelium stage to shift to the primordium formation stage. During this stage, some factor associated. One is light. Very weak light is enough to shift to the primordium stage. And the water content is 85%. And temperature is a relatively low temperature, say 15 to 25. So maximum is 20 degrees centigrade. Mycelial learning is 25 or over 27. It's very, very abundant mycelial learning occurred in the 25 or 27. But the primordium formation carried out, shifted by the low temperature, relatively low temperature, 15 to 25. This is a shift. And then after the primordium formation, some factor involve physical shock, physical shock, and low temperature. Low temperature is depend on the stream. And water supply, this factor involve, when we three factor involve, the primordium formation change to the fruiting body formation. So you can find some scheme in the bottom side. You can find the mycelial learning primordium. Primordium, so some mycelium aggregate were found in the bag or bag culture surface. Under the some shift condition, fruiting body formation stage, the primordium became to develop to the fruiting body. This is the basic fruiting process. Next slide, please. You can find this systematic development like using this slide. Slide one is primordium. This was formed on the sawdust medium. you can find some mycelial aggregate. This aggregate became too grown up. You can find the slide 2 and 3. When you can find the slide 3, the top surface of the aggregate, we can find some of the gill tissue in the this aggregate. And the slide 4 and 5, 6, you can find the top of the aggregates, mycelium aggregate, brown down to brown, and arrow indicates the gill tissue mushroom. You can find, so this process is a mushroom forming process. Next slide, please. Next, go next. Okay. This slide shows how to cultivate the shiitake bug culture. Material inoculation, incubation, fruiting, and resting. So this is the basic. The material was basically used as sawdust. In the inoculation was carried out autoclaving or high autoclaving or some boiler. The incubation was mycelial running stage. The fruiting was water supply, some proper treatment of fruiting, maintenance of the sustainable temperature, sustainable moisture, proper treatment of the fruiting were involved for the fruiting. So next slide, please. From here, I will show you the one by one the process of the mushroom cultivation. One, you can find this mixer for the sawdust mixer. Next slide, please. This slide, so like this, the sawdust were mixed by the this mixer. In this stage, we use the sawdust. Basically, oak tissue, oak wood we use, and rice bran mix, and the water content adjusts to the 65%. Next slide, please. This one, you can find this feeding media. This feeding media. This specific machine to feed the media into a bag. So you can find this baggage. You can find here the bag, polypropylene baggage. This is a very heat stable baggage. And also we set up some filter, transparent filter to atmosphere transparent. Next slide, please. This is a sterilization machine and cooling and incubation. The bag was filled with the substrate were sterilized in the autoclave. In Japan, we usually use a high pressure stream were used, but in some case, atmospheric steam were used. After the sterilization, the medium became to cooling down, and then the media were inoculated with the spawn. In this slide, automatically inoculation machine. This machine was liquid spawn they use. Next slide, please. This slide, right? Mycelial learning, you can find some bag filled and inoculated. The top of the bag, you can find the white area. The white area that now is the mycelium, shiitake mycelium, is learning, propagating, growing up. Next slide, please. After growing up, to promote fruiting, we stimulation treatment from primordium state to the fruiting state. We supply the water using water submersion. like here, you can find the sawdust medium in some bucket and then filled in the water. You can find in the right bottom side. Next slide, please. After stimulation, my fruiting, mushroom are fruiting. But I would like to here inform that in Japan we have two types of mushroom formation. One is the full face, all face, on the top right side, bottom side also, on full face cultivation. And then the bottom side you can find on the top surface. That's a mushroom fruiting point on the top side. Top side surface method, we have these two types of mushroom. Next slide, please. And this is the shiitake harvesting. This is a top surface pattern. You can find the mushroom, some farmer cultivated mushroom. So next slide, please. From here, this is a basic story of the process of shiitake cultivation. From here, I would like to inform the specific strategy for shiitake cultivation. In the case study of shiitake, this study was carried out at Tohoku area in Japan, Iwate Prefecture, especially for the Kamaishi city. Kamaishi city is, you can find in the Tohoku area. Especially this Kamaishi company, I would like to inform. This strategy carried out my friend cultivated. In Japan, basically, the mushroom farmer do not open the cultivation process because they have a specific technique to individual farmer. So basically, they do not want to open the cultivation stage. But they kindly inform me that this process. Next slide, please. Why this case study inform you? As you know, that 2011 we have a big earthquake in Tohoku area. Maybe you know. During this big earthquake, big tsunami occurred in Tohoku area. This slide shows the Iwate Kamaishi area. So big tsunami occurred in this area. So next slide, please. This is the reconstruction area 2014. The previous this place is some farmer worked and some fisher also worked. Some people were worked in here, but now no one here, no one here, no one here. So the big problem we have how to employ the people is a big problem. So they use they produce a shiitake plant in this area, a shiitake cultivation, a tsunami hit area. You can find the tsunami. Next slide, please. This is a plant. You can find some tsunami injured the harbor. You can find, and over there you can find the shiitake plant. Next slide, please. Tsunami also cause some debris. Big, so Japan house is made by rock, wood. basically wood. So the big tsunami occurred. So many debris, wood debris were occurred in the tsunami. So this is a big problem. And so next slide, please. They developed a specific boiler that produce energy from debris, tsunami occurred debris, to produce energy. This energy is used for the shiitake cultivation. This boiler is very interestingly, wet debris is acceptable. We can use the wet debris. So I hear that from the president of this company. How many boilers did you develop? So they developed 5 boilers. But interestingly, some boilers were used in Indonesia. So they wet debris were used for the energy. With debris. And next slide, please. I would like to inform the shiitake cultivation in this Kamaishi area. This is a shiitake house. Okay, next slide, please. This is a shiitake substrate. The substrate basically came from the indigenous, original wood chip. Oak tissues are used, and bran of rice bran and the mix. Next slide, please. The mixer, sawdust mixer, you can find, and the inoculation machine, and then inoculation packing machine, mycelium packing. In this plant, they produce 3 kilogram bag. And the right side is autoclave, sterilization using autoclave. And the next slide, please. This is the inoculation room. Next slide. Okay, next slide. Okay, here. You can find this inoculation room just close to the autoclave. After the autoclave, they cool down in the incubation room. And in this inoculation room, they inoculate the shiitake spawn. Next slide, please. You can find the inoculation machine in this slide. The center top, you can find the shiitake spawn. This spawn was made by sawdust spawn. And the left side is same one. Left side, the inoculation machine. The inoculation spawn was set on the top of the machine oppositely. And you can find 2 switch. One is a foot step switch, and one is a the black hip switch. So only one person can carry out the 2 steps. One is firstly step switch, mixing the spawn and filling in the inoculate. And the second switch is a sealer. You can find over there, heat sealer. After 2 steps, after inoculation, sealing. All 2 steps is carried out, only one person. Next slide, please. This one, the incubation. Then firstly, they culture the shiitake. Firstly, the low temperature at 18 degrees. Firstly, the slowly culture and myself learning start. And the next slide, please. After mycelium fully colonized, some water is produced. Maybe this came from the respiration of fungi. So we should avoid to use this water. So to avoid use water, we change the position of the cultivation bucket. Next slide, please. In this plant, they produce high quality mushroom on the top surface. But so they wanted to inhibit the small mushroom. So they inhibit to produce the shiitake side and bottom. So they want to inactivate the primordium by heating and no oxygen treatment. For in the summer, you can find in the right side polypropylene bucket filled with relatively hot water, 27 to 28 degrees in summer condition, for 7 to 10 days. So they suppressing for the fruiting from the bottom side and side. This is a killing stage for the inactivation stage. Next slide, please. So, in this system, they open the bucket on the top, not on the top. Side and bottom side is not open. On the top of the bucket are open. They and then produce fruiting body on the top side. Next slide, please. After harvesting the mushroom, the substrate were resting for the next fruiting. And during the next fruiting, when we finish the resting, they produce a second time flushing by using water supply. In some case, the left side, you can find in the some farmer supplied water by spray. Next slide, please. This is the post-harvesting process. After harvesting the shiitake, they selected the grade and packaged and sealed. This is a post-harvesting process. Next slide, please. Here, I would like to summarize the characteristics of agri-kamaishi strategy. One is the use tsunami hit area. This means employment of kamaishi area. Some farmer, they have no work, so they provide some work using mushroom cultivation. And the second characteristic is the specific boiler acceptable for the water debris acceptable. But this specific boiler is acceptable for the user. Mushroom after cultivation, we produce waste substrate, use the substrate. They use substrate also used for the boiler. And the second characteristic is fruiting is limited on the top surface. This results in the high quality of mushroom. This is a characteristic of this area. So next slide, please. I will show you another case study of maitake. Maybe maitake mushroom is not familiar in Indonesia area. But in Japan is also they like a very much maitake. So I would like to inform you how to cultivate maitake. Maitake is a relatively very weak mushroom against fungi, pathogenic fungi, very weak. Trichoderma attacked easily to maitake bag. So I would like to show you how to maitake cultivate. This maitake was cultivated Shimane Prefecture. Shimane Prefecture is just close the Tottori Prefecture. Our university is Tottori, just close to the Shimane. Here, I will inform the maitake cultivation. Next slide, please. The maitake. This maitake, this you can find the maitake mushroom. This cultivated this farmer, very big maitake mushroom. They cultivate. They have a specific know-how to cultivate this big maitake. This maitake was after cultivation. This mushroom is easily. Some Japanese people desire to use this big mushroom. So all of the mushroom easily sold directly, not supermarket. Next slide, please. Sorry, this is the process of written in the Japanese. I will skip this slide. Next slide, please. This farmer, they use sodas, local sodas, sign-in area, local sodas. And they use wheat bran. Wheat bran are very difficult to purchase in Japan, so they came from the another prefecture. They use this local sodas. Next slide, please. They also use sawdust medium. The volume is 3 kilogram sawdust. They use a transparent polypropylene bag. After the important point is after the sterilization. After sterilization, they significantly slowly, normal pressure, very slowly, 1 day, 1 night, 2 days, because to avoid make a space between the baggage and substance. When we quickly normal pressure return to some spaces opened between the bag and substance. This is difficult to make a big mushroom. They want to produce maitake mushroom only top and special point. So they carefully after the sterilization, they carefully move to the normal pressure. Next slide, please. And this is a mycelial running stage. During the mycelial running stage, they make improve the holding to make open one stage to how to fold. They have a specific holding style. And during this stage, the attention point, but careful movement, they do not do rush movement. It's very difficult. When we rush movement, some mushroom were produced on the side point とかね。So they carefully move. Very, very carefully movement they require. After the mycelium, next slide please. You can find the fungal mass on the top of the bag. This black point is the fungal mass of maitake. So if we have an open space between the bag and substrate, this fungal mass is homed on the side. So it is very difficult to make a big mushroom. So they want to produce fungal mass on the one top open this space. Next slide, please. This is fungal mass producing process. Next slide, please. After the fungal mass was developed, after the fungal mass, they waiting for low temperature. The fruiting on the one space will produce one bundled fruiting. This bundled fruit, Japanese name is 一株舞茸ですね。株舞茸。Next slide, please. During the cultivation, next slide, please. Oh, okay. During the cultivation, they have monitored the temperature, relative humidity, and carbon dioxide control carefully, because the maitake is very carefully control the CO2 level. When we high temperature, high CO2 concentration induce abnormal maitake formation. So they carefully check the carbon dioxide at condition. Next slide, please. After the cultivation, they collect the substrate and harvest the maitake like this. Some farmer collecting the maitake. Next slide, please. After the collecting the maitake, what sawdust do not use. Only one time. Only one time. No second flushing. Now, next slide, please. And then after the harvesting, the culture mushroom were directly sold in this area and some internet area. Next slide, please. Next slide, after the used substrate, what's the substrate were the selected bagged sawdust. Next slide, please. The one is a bag, one is a sawdust. So carefully selected the sawdust and bag. The sawdust was used as a fertilizer, composting and used for the fertilizer. Hey, next slide, please. So here I would like to summarize the characteristic of this Okuizmo maitake. One is the use of local sawdust as a substrate, and fruiting is limited only one open space. After the second point, after sterilization, slowly movement, slowly normal pressure is the key point to make a big mushroom. And the careful movement of the substrate is also the second key point. And selecting bagged and substrate and the direct selling is some of the characteristics. So this is some characteristic of Okuizum Maitake. So next slide is my recent work. So I would like to skip this process because of time limitation. Yeah. Oh.
10 minutes.
Eh?
You still have 10 minutes. 10 minutes.
10 minutes?
Yeah.
I can use only time. Is it okay? I can continue. So I would like to briefly introduce my strategy for the shiitake. My ectomycorrhizal mushroom, show myself. But this edible mushroom in Japan as a miso soup. You can find in the miso soup here. This mushroom is closely associated with the black pine tree. Next slide, please. You can find ectomycorrhiza. You can find here. This is the fungi very associated with pine root. Next slide, please. This is a symbiotic relationship. No need to. I would like to skip. Next slide. In this strategy, I wanted to make some specific spawn for ectomycorrhiza mushroom. So, because some when ectomycorrhiza mushroom are cultivated aseptic condition, we can cultivate mushroom, but aseptic condition is very difficult because high cost is required. Then the spore suspension also can use for the inoculation, but it's not a practical method to cultivate. So I wanted to make, next slide please. I wanted to make a specific spawn to cultivate, like a spawn. Next slide please. Next slide please. Next slide. Okay. So firstly, you can find the significant difference of the strain. One is the low colonization strain, right side high colonization strain. We selected firstly to the useful strain of Sholo. Next slide, please. And I found the two strain is very useful, but the when we inoculate, after 2 weeks later, quick inoculation, homing of mycorrhiza homed in the two strain, number 4 and number 10. Using, next slide, please. Using this strain, we have make mycelial slurry, homogenizing slurry. Submerged culture, we homogenize. But homogenization, we use a chopping machine, not homogenizing, just generation to avoid physical damage to the mycelium, chopping machine. And artificially inoculated, injected to the seedling. Next slide, please. And I also use surfactant to the inoculation detergent, 280. This is small amount of 280 is very useful to stimulate mycorrhization. Next slide, please. I also use the mother plant system. This mother plant system is used for the truffe mushroom in French, Italy. So I also use this system. Next slide, please. Then after the mother plant system, we artificially inoculated in the July and September only 2 times. And next slide, please. This slide shows that you can find the significant different from the uninoculated and the right side they inoculated twice. Move back. Uninoculated means the only mother plant system. Only mother plant system is also produce microlide, but artificially injected plant, we can find many microlide here. So next slide, please. So interestingly, very surprisingly for me, this show mycelium or ochre produce this inoculated ear. This is very surprising for me. Okay, next slide, please. Well, only mother plant system we can find only one mushroom, but the artificially inoculated twice we can find 8 mushrooms. Okay. 2 minutes? Yeah. 2 minutes? 2 minutes. So I would like to inform another outplanting the fresh, outplanting of the field. The interesting, next slide, please. Outplanting, when we outplanting, we carry that concentrated outplanting, because when in the outplanted too, you can find that many microorganisms in the outside. When we one by one separated inoculation, transplanting, maybe the to keep a dominant condition to biomass, we gather concentrated plantation carried out, for example, 3 by 3, 25, 4 by 4, and so on. So interestingly, when we, next slide, please. Very interestingly, we found many, when we concentrate plantation, produce a quick mushroom production. Next slide, please. Next slide, please. Next slide, please. Next slide, please. Oh, okay. When we carry that concentrate plantation, we have found a mushroom. But solo only 1 or 2 seedlings is difficult to produce a mushroom. Okay. Next slide, please. This is final. The 2 years old nursery, we can many mushrooms produce here. Next slide, please. This is a cultivated short mushroom. Next slide, please. This is a cultivation history, cultivation process. Next slide, please. This is a summary of my study. Next slide, please. Next slide. That is all I have to say. Thank you very much. So finally, I would like to say all of you, I am hoping that you can deepen the understanding of the mushroom biology. I also hoping that you have obtained a very useful information, valuable information of mushroom cultivation. And finally, I would like to hoping that you have my lecture is contributed to your specific scientific life. Thank you very much.
Okay. Thank you, Professor Shimomura. Please give applause to him. Okay, we still have 15 minutes for discussion. And then I just make the summary of this Professor Shimomura presentation today. This is about the 3 kinds of mushroom cultivation. The 2 of mushroom is shiitake and maitake is saprophyte mushroom. And the 3rd one is soro, in Japanese is soro, is a mycorrhiza mushroom. So it's a little bit different in the cultivation method. Okay, we are open discussion from the participant in this room. You can raise your hand. Okay, please say your name and then the faculty. Okay.
Hello, good morning. My name is Shania. I am from Faculty of Science and Technology from biology major. So I have a question. I'm interested when you show us the wood cultivation figure. And so I have a question that in Indonesia, mushrooms are commonly cultivated in sawdust-based substrate and then put it in a plastic bags. And if we compare those method with traditional log cultivation, do you think there is a potential advantage and disadvantage of this method, particularly in terms of the mushroom quality and the bioactive compound itself? Thank you.
Okay, Sensei, it's your time to answer this question.
Okay. Thank you very much. A very, very interesting question to the different for the cultivation style, wood log and sawdust cultivation. So I firstly wanted to say that the quality of mushroom is quite different from the wood log and sawdust. But recently, the difference is became too small because high quality shiitake mushroom were produced by using a top surface cultivation method. They produce a high quality of mushroom. So quality is very, very close to the wood log. But wood log is a very tasty texture, very quite different from sawdust medium. So very different. But recently it's very resembling. So in Japan is also the traditional method is carried out very, very old farmer. But now recent farmer, a young farmer, they wanted to use the wood log because very heavy. Bag is only 3 kilograms, very light. And also they can do not use the forest, house, planting. So they recently, young farmer tried to cultivate the shiitake sawdust medium. And the second point you asked me to the some medical compound difference. So I'm not sure. But some compound are very different. Some compound, but all of the recent active compound were produced by the sawdust-based method, shiitake, maitake also. But few data is available in the wood log cultivated shiitake, maitake also. This is my answer. This question is acceptable for you?
So, is it okay?
Yeah, thank you.
It's okay. So, Sensei, how about the time for harvesting? Which one is faster? Using the backlog or using the wood log?
Yeah, especially wood log is very. Wood log is require many time.
Wow.
At least when we 1 year, 2 year is required to machine learning. But bag culture is very quick.
Yeah,
relatively only 3 months in shiitake.
3 months.
Shiitake. But another mushroom, for example, oyster mushroom, 1 month, it's very, very quick. But wood log shiitake is very high, long time required to fruit.
Okay, thank you. Okay, is there any question from the audience, please? State your name.
Thank you very much. To be honored. My name is Maya Eva. I have a question, but first of all, I wanted to say I took Japan have an amazing idea about to remake tsunami area to be confined mushroom. But I have some question. Is it safety to production the mushroom in there?
Okay, once more. Okay, once more. Once more your question. Okay.
Japan have amazing. Japan have a place to remake mushroom convention after tsunami.
Yeah.
My question is it a safety to production in there?
Yeah. How about the safety in the tsunami area? Yeah.
Of course, maybe someone very worried about the radioactive material absorption by the shiitake. But all of material already checked by the very severe checking process. So they give supplied for very safety material. But some they wanted to warning about the after harvesting. So harvesting material, the substrate, the wood log, wood chip, some wood radioactive material contains some wood chip. So some radioactive cesium concentrated to the shiitake. But to use the radioactive, to use the substrate, we already checked for the radioactive material. So especially this is at Kamaishi area, Tohoku, Iwate Prefecture. There is no radioactive contaminant in here. But in some case in the Fukushima area, some remaining for the radioactive contaminant. But the Kamaishi area is no contaminant. But so anyone very worrying about this? I understand that you want to say that.
Yeah. Okay.
Thank you very much.
Okay, thank you. Yeah, because in Tohoku area, the earthquake not hit in the area that have the nuclear power, Sensei, right? But the another when you have the nuclear power, so we worry about the radioactive contaminant in the substrate. Yeah. Okay. Thank you. Is there any question here? Okay, please.
Good afternoon, Mr. and good afternoon, everyone. My name is Jabira. Thank you for the interesting presentation. I have a question. Considering the increasing of sawdust and polypropylene bags in mushroom qualification, how can we increase mushroom production while reducing environmental impacts and contamination risks? Thank you.
You mean the environmental effect when we use the sawdust as a substrate or something like that? Once more.
Considering the increasing use of sawdust and polypropylene bags in mushroom qualification, how can we increase mushroom production while reducing environmental impacts and contamination risks?
Okay, you mean how the environmental effect when we use the sawdust and plastic as a substrate in the mushroom cultivation, Sensei. Okay, thank you.
I understand that you wanted your question that but why most of the big problem is the bag, polypropylene bag. It's a very insoluble to this is not the use for that polypropylene bag. This is a big problem. But now we are to use how to disorganize this propylene bag. It's a big problem. But not only remaining now, sorry. And the another problem is the used sawdust material. Sawdust material is also a big problem in Japan. So they mainly used for as a compost and as fertilizer to produce some vegetable. So as a fertilizer. And recently, we have a very interesting data from how to use the substrate, because this substrate is antifungal compound were contained, so very big biomass to produce antifungal materials. So they wanted to use waste substrate to control the pathogenic fungi and to cultivate some rice, vegetable, to how to cultivate, and during a cultivation, what's the substrate? Now we try to use what's the substrate, shiitake, another mushroom. It is a bit now undergoing to how to use what's the substrate. Thank you very much for your question.
Okay, thank you. We still have 3 minutes. I think from the online participant, is there any question? No. Okay, there is no question from the maybe in the right hand. Okay, cannot see. This one, if the earth starts to become polluted and plants cannot grow and livestock cannot no longer survive, will mushroom be able to replace the source of protein and human needs in the future? This one, Sensei. If the earth starts to become polluted and plants cannot grow and livestock can no longer survive, will mushroom be able to replace the source of protein and human needs in the future? Can you read it, Sensei? So this one, Sensei. Okay. It is possible that mushroom be able to replace the source of protein and human needs in the future.
This question is a very big question to how to use the mushroom. Maybe you know that mushroom contain specific compound. Maybe next speaker have many useful information. Maybe you know that. The most very interesting point is that I'm very focusing on the protein production by using protein production mushroom. And mushroom contain about 3 to 5% of the protein. This is converting from the protein. The original material is a waste substrate, solid. So not useful material originated from converted to the protein. It's a very, very beneficial point of the mushroom. So I believe that this mushroom conversion from some waste substrate to the protein will be evaluated more in near future. It is my opinion.
Okay, thank you, Professor Shimomura. Okay, because this time is limited, we have to finish this discussion, and then thank you very much for Professor Norihiro Shimomura for your valuable presentation today, and then please give applause to him.
Thank you very much. Thank you very much. Terima kasih.
Terima kasih. Now time is for you. and the OMC.
Okay, so let's give another round of applause to our speaker, Professor Shimomura, and also our moderator, Dr. Iwan Saskiawan, who already finishing their first presentation. And thank you once again, Dr. Iwan and also Professor Shimomura. You can have your seat again. But now, I would like to inform to all participants who joining us in this visiting professor session, don't forget to fill the attendance form that already shared by the committee at the chat box of the Zoom meeting. You can have your certificate of participation by filling the attendance form that already shared by the committee at the chat box of the Zoom. So, after having the first presentation session from Professor Shimomura, next we will have our second visiting professor session with the second presentation that will be delivered by our second presenter or speaker, that is Professor Tsuyoshi Ichiyanagi. And also this session will be guided or will be moderated by one of the lecturer that coming from the Faculty of Science and Technology of Universitas Terbuka, especially from Biology Study Program. So, ladies and gentlemen, please join me in welcoming Mrs. Hikmah Zikriani, MSE.
Tes. Okay, thank you so much everyone, and thank you MC Miss Afira for introducing me. So, Assalamualaikum warahmatullahi wabarakatuh. So, good morning everyone. Okay, how are you today? Okay, I hope today you still have an energy because we have another speaker that will be there will be good also for from the Faculty of the Agriculture, Tottori University. First of all, with sincere respect, I would like to begin the session and welcoming everyone who is present in this room and also joining us online today. So first is our honorable speaker, Professor Norihiro Shimomura, and also Professor Tsuyoshi Ichiyanagi from the Faculty of Agriculture, Tottori University, Japan. And then the Dean of the FST UT, maybe she will be joining us in online. And then the Vice Deans of FST UT, Dr. Sita Ali Farihati, Master of Science, Dr. Anda Inggit Winarni, Master of Science, and Dr. Pepi Rospina Pertiwi, Master of Science. To all head study program, especially biology study program, Dr. Elizabeth Novi Kusumaningrum, Master of Science, and also Dr. Rini Riviani and Dr. Iwan Saskiawan from RC for Applied Microbiology, Brain Cibinong. And also not to forget, to all participants, lecturers, colleagues, staff, and also to the students who are joining us, I've seen here with the yellow alma mater. Welcome everyone, and also joining us in the online. So it's very warm welcome to all of you. Okay, so my name is Hikmah Zikriani, and today it's really an honor for me to standing in here. And you know, I supposed to be like introducing an amazing speaker today. But before it, I think I want to have a little bit chit chat with you all. Okay, is it okay? Is it okay? Am I too fast? Okay. Okay, I'm a little bit slow down. So before I am welcoming the second speaker, I think I would like to have a little bit like a communication to all participants to make sure that all of you ready, because we still have one more amazing speaker for today. So after hearing the development of the mushroom science in Japan from Professor Shimomura, so let me ask you one question. So what do you think is the next step after we understand the mushroom better? So what we're gonna be learn after we understand about the mushroom, and then what do you expect more from the mushroom? In your food, maybe for your healthy, maybe for everything? Okay, can we produce the mushroom? Okay, for what? Is it only for food? Or do we expect more from the food we eat from a mushroom? Yes? Okay. Okay. Do you know why? Because this amazing speaker today, he will be presenting to us about the organic chemistry, synthetic and also natural products from a mushroom. Is that true? So it's a really pleasure to me to introduce us to the Professor Tsuyoshi Ichiyanagi from the Faculty of Agriculture, Tottori University, Japan. Please joining us. So Professor Tsuyoshi Ichiyanagi is a professor of the Faculty of Agriculture, Tottori University, Japan, and has served as a director of the Fungus Mushroom Resource and Research Center since 2019. Okay. Say, can you please help with? Okay, wait, okay. Okay, so let me repeat. So Dr. Tsuyoshi Ichiyanagi is a lecturer from the Faculty of Agriculture, Tottori University, Japan. So he is a professor and has served as a director of the fungus or also mushroom resource at the research center since 2019. So he received his doctorate engineering from University and has been actively involved in the research on the organic synthetic chemistry and also bioactive compounds, particularly the isolation, structural determination, and also chemical synthesis for the bioactive compound from a mushroom. So, good morning, Professor.
Good morning.
Okay, so are you ready?
Yeah.
Okay, so your time will be the same as Professor Shimomura, 45 minutes for your presentation. So I'm really sorry if I'm gonna remind you about like a 5 minutes before the presentation is end. Is it okay for you?
Yeah,
okay,
it's right,
and also we have 15 minutes for the Q&A section after you finish with the presentation. Okay, so everyone, please give, wait a minute, okay. So please everyone give warm round of applause for the Professor Tsuyoshi Ichiyanagi from Faculty of Agriculture, Tottori University, Japan. So Professor, the place is yours.
Good morning, everyone. And my name is Tsuyoshi Ichiyanagi from Tottori University. So first, I'd like to thank Ma and Dr. Yuan and the organizers for inviting me here and giving me the opportunity to present our research center and our project. Today, I'd like to introduce the outline of the FMRC, the Fungus Mushroom Resource Research Center, and our recent research project. So, I am now very nervous, so I'm sorry. Yeah, my English is not my native language, so please bear with me if my pronunciation is not perfect. So I will be check my note occasionally, but I will do my best to communicate clearly. So. Okay. So here is an overview of today's lecture. First, I briefly introduce the FMRC, the organization I'm affiliated with. Then I talk about the FMRC's three main areas of the activity. The first is collection, preservation, and provision as genetic resources. The second is the discovery of bioactive compounds from fungal secondary metabolites. The third is a chemical synthesis of bioactive compounds, which is my area of expertise. It's a small time, so it's a skip. Sorry. Yes. Okay. FMRC was established in 2005 and has two distinctive features. First, it is a unique institute among the universities in Japan that focus on the education and research of mushroom sciences. Second, FMRC has the world biggest culture collection of mushroom strains. These two is a feature of our center. FMRC is divided into two research divisions. One is basic science that focuses on the taxonomy, phylogeny, molecular genetics, and the conservation and the characterization of the strains. The second is applied division, which focuses on the cultivation science, natural product chemistry, food science, pharmacology, and environmental remediation. I'd like to present today the outline of these divisions. This is our purpose and the mission. Mushrooms are well known for their diverse biological properties. Their use as food and medicine, while some species also known to be poisonous. However, there are still many mushroom species whose biological properties remain unexplored. As FMRC, we focus on the mushrooms and aim to develop human resources who can contribute to discovery and utilization of new fungal resources through systematic education in fungal resource science. We also aim to contribute to the vitalization of existing industries and creation of new industries by promoting high-quality research. Since 2012, the FMRC has provided mushroom resources to researchers for scientific research. In addition, with support from next Japanese government, we have been working since 2022 to establish the research center dedicated to the collection, preservation, and the provision of the mushroom resources. Okay. Now, let me give you an overview of some of the research and activities of FMRC. The starts are collection, the preservation, and the provision of genetic resources. Okay. And as I mentioned earlier, one of the FMRC's main activity is collection, preservation, and the provision as a genetic resource for the research. Yes. These activities are carried out by basic science division. Our work can be broadly divided into 5 areas, so it's shown here. Now let us close look at what we actually do in each of these areas. The starting point of our research is the collection and the taxonomic study of mushrooms. First, we go into the field and collect mushroom specimens. Okay. Then we identify and classify them based on their morphology and then establish living cultures from collected specimens. Currently, we are focused on the genus Agarix and Corticeales. Through these studies, we aim to discover new species and better understand their diversity. This slide shows how we identified and classified mushrooms at FMRC. As you know, mushrooms are we see in nature and genetically called fruiting bodies. First, we observed morphological features such as the cap, the juice, and the stem using the naked eye and microscopy. Next, we examine their spores and mycelia using electron microscopy. Finally, we construct a phylogenetic tree based on DNA sequence data and compare the results with existing data to identify the species. Okay. When we discover a new species, we give it a scientific name. Yeah. These photographs show some examples of new mushroom species that have been described by researchers at FMRC. It is established that there are more than 1.5 million fungal species on Earth, including approximately 5,000 to 6,000 species in Japan. However, only a small percentage of these species have been formally described and given scientific names. This means that there are still many species waiting to be discovered. In other words, there is still a great deal to explore in the world of mushrooms. As a FMRC, we used 2 methods to obtain the cultures from mushroom specimens as shown in this slide. The first is polyspore isolation. We attach a spore-producing part of mushroom, such as juice or pores, to the lid of Petri dish using Vaseline. The spores are then released and germinate, producing hyphae on the agar medium. The second method is tissue isolation. The method is used when the spore of mushrooms do not germinate well on the agar medium. In this case, we cut a small piece of tissue from inside the fruiting body and transfer onto the agar medium. The mycelium then grow out from the tissue block. Using this method, we can establish mushroom culture strain for preservation, further research. Once the mycelium has been cultured, it is transferred into the cryotube using the straw, like this. The strains are then frozen using a programmable freezer, here. Then the most strains, such as saprophic fungi, we use agar medium for cryopreservation. For strains that are more difficult to preserve, such as mycorrhizal mushrooms, we use a porous culture media instead of the agar media. Yeah. Then mushroom, so how many strains do we actually have? Currently about 8,700 strains are preserved at FMRC. We verify their species identities using DNA sequence data, mainly from the ITS lesion of ribosomal DNA. Of the about 2,300 strains, quality-controlled strains are currently available to researchers for distribution. This slide shows the search system that allows researchers to find strains that have undergone quality control and available for distribution. The search system can be easily accessed through the FMRC website here. Researchers can search for strains using various criteria, including the TFC number, a scientific name, the Japanese name, and keywords. It also provides the homology search function, allowing the user to search for strains based on DNA sequence similarity here. So researchers can easily find the strain they need using the variety of search options. This is an example of an individual strain page. Researchers can find detailed information about each strain, including substrate, collection history, and characteristics. They can also view photographs of the strain growth on the culture media here. Small one, sorry. Then in addition, DNA sequence data that are available for the confirmation. That concludes the overview of our activity related to the collection, preservation, and provision of genetic resources. So next, I'd like to introduce some of the research activities carried out applied research divisions. These include research on mushroom cultivation and breeding, the discovery of bioactive compounds produced by mushrooms, fruiting bodies, and mycelia, and chemical synthesis of these compounds. So these research activities are carried out by Applied Science Division. In recent years, we have been focused on several research topics. One is development of cultivating method. The second is improve crop varieties. Another area is so we search for bioactive compounds, the development of pharmaceuticals, pesticides, dietary foods, and healthcare products. Okay, so today I focus on my research on bioactive compounds produced by mushroom and their potential application in pharmaceuticals, pesticides, and functional foods, and others. So, as you know, mushrooms have long been history of use as a traditional medicine. For example, Polycoccus and Polyporus and Ambratis have traditionally been used as diuretics. So other mushrooms, including the shiitake or ganoderma and the tramera, have also been shown have a wide range of biological activities. These activities are associated with various bioactive compounds such as terpene and polysaccharides, including beta-glucan. This makes mushroom promising resource for drug discovery. However, we still lack systematic information about their biological activities. One reason is that it's difficult to collect and compare a large number of quality-controlled strains at the same time. This slide shows the main challenge we face when searching for bioactive compounds for mushroom, as well as how we address these challenges at FMRC. There are 5 major bottlenecks in this type of research. The first, screening for bioactivity requires access to a diverse range of mushroom species. The second, many wild mushroom strains have not yet been identified at the species levels. The third, a large quantity of fruiting body is often required to isolate and characterize bioactive compounds from single species. Fourth, large-scale cultivation facilities and advanced analytical equipment are essential for this type of research. And fifth, because process requires considerable time and effort, it is difficult for the individual researchers to carry out all of these steps alone. So how do we address these challenges? The FMRC has diverse collection of mushroom strains that have been identified at the species level. In addition, we have team of the experts in taxonomy, mushroom cultivation, natural product chemistry, analytical chemistry, medicine, and veterinary medicine, and chemical synthesis. By bringing together these resources and expertise, we aim to develop high-throughput screening platform and establish methods for the rapid isolation and identification of bioactive compounds. Our goal is to uncover the potential of mushrooms as bioresources by comprehensively exploring their unique bioactive compounds. To do this, we prepare extract from our mushroom strains, build an extract library, and simulationary screening using multiple bioassays. By screening this library, we aim to discover novel compounds with unique biological activities. Yes. Now I'd like to explain how prepared our mushroom extracts using the next 2 slides. Mushroom extracts are prepared from both fruiting bodies and mycelia using a variety of solvents. This slide showed an example of how we prepared an extract from mushroom fruiting body. First, wild or cultivated fruiting body is freeze-dried and ground into the powder. The powder is then sequentially extracted with organic solvents, such as ether, and dichloromethane, and methanol. So after extraction, the mixture is filtered and the filtrate is concentrated. The remaining materials is then further extracted with water and concentrated, obtained the final extract sample. So in this way, we obtain the extract sample containing a wide range of compounds with different chemical properties. So this is a preparation of mycelial extract. First, mycelium is cultured in a liquid medium. The culture then separated into the mycelium and filtrate. The mycelium is freeze-dried and crushed and extracted by methanol, then concentrated to the extract, mycelial extract. The filtrate is extracted with ethyl acetate, organic solvent, then concentrate to make the extract. In this way, we have prepared a library about 2,300 mushroom extract now. So this extract library enabled us to screen a large number of samples simultaneously, allowing us to efficiently identify the mushroom species that produce bioactive compounds. Okay. This table showed some results of our screening. And the first, this column is shows a total number of extracts tested, while the other this area column showed the number of extracts that exhibit activity. As you can see, many extracts showed a positive activity against a variety of targets, including bacteria, fungi, the virus, parasite, and cancer cells. So, however, the number and the strength of active extract varies depending on the target. By continuing this screening process, we expected to identify more mushroom species with promising biological activities. So, the next step of our research, the isolation of identification of bioactive compounds from extract that showed biological activity in our screening. So I will focus on the two recent examples. The one is mode inhibitor targeting the fungi commonly found in air conditioning system, and a compound with anti-malaria activity. Yeah, I'll explain our research process for the isolation and identification of bioactive compounds. So first, we cultivate mushrooms selected through our screening process. As FMRC, we usually start with about 5 liters of mycelial culture. When we use fruiting bodies, we directly extract them with hot water or organic solvents. So if the active compounds are lyophilic, we perform the liquid extraction of the culture broth using the organic solvent. The solvent is then removed under the reduced pressure to obtain the crude product. Then we separate the compounds. Volatile compounds can be separated by distillation, while non-volatile compounds are usually separated by chromatography. In some cases, we also use crystallization. Each fraction is then tested by biological activity and using the bioassay guided fractionation until the active compound is purified. So further separation, we use different types of chromatography depending on the property of the compounds. So after the purification, we get a single compound. We used many analytical experiment by mass spectra or NMR study. So finally, we performed a further analysis determined its stereochemical structures. So now, first, okay. Yes, first I'd like to talk about search for antifungal compounds against mold that grows in air conditioning system. So I think everyone is familiar with air conditioners. So air conditioners are used for a long period. So molds can start growth about outlet here or inside here. So it's a problem. So the molds can cause unpleasant moss odors and in more serious cause many contribute to respiratory problems. To address this problem, we are searching compounds produced by mushrooms by using the mushroom extract. So this is screening system. So this slide is skip. I'm sorry. So skip. We so tested the results. So okay. Please show the photo. So the top of the side is a start of the screening. Then lower shows a plate after 2 days of the incubation. The image of left here, so no visible change the start from the photo, so indicating that fungal growth was strongly inhibited. So the middle is image weak inhibitor. The right side is not inhibition. It's okay. So we tested about over 1,500 samples, tested mushroom extract. We found 3 or 8% of extracts showed growth inhibitory activity against the molds. Among them, about 1 or 2% showed a strong growth inhibitory activity. Oh, thank you. So this table showed more strong results inhibition of the mold production. So then among them, so we focused on the Glyophyllum species and the target compound, sample to isolate the bioactive compounds. So this is strategy of the isolation scheme. So first we cultivate, extract from the fruiting body to get the extract. Then separate by silica gel chromatography. Then so more than tested under separation step. So to get we get 4 compounds. So this is data out of the compound data. So it's skipped and finally so we determine the analysis of 2D NMR spectrum, so to determine the structure here. So the same manner, so we also determine the 4 compound structures here. So and then we synthesize this compound by using rhodium catalyst, and we can get many gram scale of the target compound we can synthesize. So the last topic is so anti-malaria compounds. So this is well known, the malaria cycle, life cycle here. So these three compounds are missing. That chloroquine is well known, the strong medicine we use, but also this drug-resistant malaria is appears. So we have to develop the new type of malaria medicine. So we try to the new compound from the mushroom extract. So we collaborate Dr. Ito and Otsuki and their faculty of medicine. So they assay the screening system about this one, right? So then we find 51 samples of culture medium extract and the floating body extract, 39 samples is now over 90% inhibit data. So we target by the appropriate this type of mushroom to get anti-malaria compounds. This figure, the scheme is also isolation system. So we got 3 compounds and determine the chemical structure here, and finally, so we get speciosin B as a target compound. However, so speciosin B is a mixture of the diastereomers. It is to clarify the diastereomers, we have to synthesize the chemical synthesis of these compounds individually. So the scheme is skipped on the here. So finally, so we finish the total chemical synthesis of diastereomers individually. So R form and S form. So compared with the natural product data, top was is so natural product data. They compare the middle and the bottom is synthesized compounds, both diastereomers. Please see here, so natural product mixture of diastereomers here, but so synthesized is very clear. So this data show the natural product is a mixture of diastereomers. It is a first findings of the speciosin B synthesized. Okay, this is summary of the natural product chemistry. So after, I'm sorry, just give me 1 second. These are these compounds, as we found recently, determined bioactive compounds at FMRC. So these compounds, so for example, so inhibition of the circadian rhythm regulation, antibacterial activity, tyrosinase inhibition, anti-malaria, and the plant growth promotion. Very wide range of bioactivity in the these compounds. So, okay, so sorry. So this is final slide. So our center contribute so many professors here, and today anti-malaria compounds and anti-mold compounds. So the examined is by using the many students. So I'd like to thank these members, and finally thank you for kind attention. Sorry.
Okay, thank you so much, Professor. And please, everyone, give one applause for Professor Ichiyanagi for amazing. Okay, so I'm gonna open a Q&A session. So if anyone in here wants to ask a question to Professor, please raise your hand. Oh, okay. Thank you. Okay. Please give your name and also from
Thank you. Good morning, Prof. Good morning, everyone. My name is Rira Harjo, and I am a biology student at University Terbuka. My question is in the short for bioactive compound production by mushroom. What are the biggest challenges in the identified compound that have real potential for pharmaceutical of medicine application? And with approach do you think are most promising to overcome this challenge? Thank you.
Okay, so your question is asking for the challenge. Okay, what is your challenge in founding the bioactive compound from a mushroom, and how do you handle those challenge? Yeah, the challenge is more like the difficulties you have during searching for the bioactive compound in mushroom, and how do you handle those challenge?
Thank you, the comment. So in my field, so I'm challenged. My challenge is that how to construct the compound structures to develop the pharmaceuticals. Okay, and production this is difficult. So our final goal is new pharmaceuticals development. So this is very challenging theme. So I could not. It is difficult to myself to make the pharmaceutical. So we have to make that team. Then the role of the taxonomy and production, isolation, structure determination, on the chemical synthesis part, expert contribute team. So my challenge is chemical synthesize, yes, that to make the chemical structure by using the easy the technique and
Okay, do you understand? So it's more like creating the chemical synthetics for the bioactive founded for the pharmaceutical purpose. It's the challenge actually because it is going to take a week for synthesizing itself. So is it clear for the answer? Okay, thank you so much. And then another question. Oh, okay. Please mention your name and the question.
Yeah, hello, good afternoon. I am Sania from Faculty of Science and Technology. I have a question. First of all, thank you for the interesting presentation. So I've always been particularly curious in mycelium-based biomaterials, and in recent news, I don't know if it is recent enough. It is said that mycelium-based biomaterials, it has a potential to support human mesenchymal stem cells. So my question is, do you think mycelium-based materials could combine structural support with therapeutic or drug delivery functions? And what are the main challenges in bringing this concept into practical biomedical application? That is, thank you.
So, okay, so the question is talking about the mycelial growth. It stated that it can be somehow promising to the stem cell. Yeah, it's a part of the stem cell. So is it really possible for that? Is it true, the question?
And what are the main challenges of bringing this concept into the practical biomedical?
Okay, and is it like another maybe a challenge bringing this concept like the mycelial itself for the stem cell, and then what could be a challenge, and then bringing those concepts into practical, if it's really possible. Is it hard?
Yeah. It is a difficult question for me, so different.
Okay.
Yeah, so I'm sorry, so I could not so answer to you. So I asked another professor so to get the back to your answers. Is it okay? I'm sorry.
Oh yeah, it's okay. Thank you. Thank you.
It's okay, Professor. Okay, I think there another, I think one more question. Maybe I can ask in the Zoom. If we have a question, please use a chat box. Oh, we already have it, Professor. So there is a question from Nico. Okay, is it okay if I read it? So are the production and composition of bioactive compounds stable in fungi cultivation across multiple generations? Yeah. The production and composition of the bioactive compound, is it stable like in one fungi for multiple generations, or is it going to be like a change? Okay. And also for the subculturing or genetic rejuvenation generally leads to the genetic stability. And the industrial level, are there any recommendations or practical methods to ensure consistent stability and production of the bioactive compound? So it's asking for the generation to generate, like for creating the offspring and another offspring. How do we create like a stable bioactive compound?
So I think we can control the bioactive control production. But I'm not so difficult to the technique or the strategy. So I have no idea.
So there is no like giving or any recommendation for the practical methods, but usually for over generation, is it going to be stable? Like for the example, the species of the oyster mushroom, if we cultivated it and then reculture again, is it for the bioactive compound will be stable for over generation, Professor?
Sometimes it is okay. So sometimes it's difficult. Sometimes okay.
Okay, so
it is depend on the conditions of source.
Okay, so it depends on the condition or where the mushroom growed up. So usually it's stable, but depends on the condition, right? Okay, so it is gonna be like the reason why maybe it's stable, but there is also a challenge in that part. Okay, thank you so much. So I think I'm gonna open one more question, Professor, in the chat box. It is from Muhammad Muzahid. It's from FST UT Biology Study Program. How do the bioactive compounds derived from mushrooms interact with a cellular signaling pathway to modulate immune response? And what is the implication? Do these mechanisms have to be development for the novel therapeutical agents against the chronic disease maybe? So I think it's related with the pharmaceutical, because I think the world is still looking the drugs or the curing for the cancer. How can it be derived from a mushroom?
Yeah, it is okay.
Yeah, you can answer that. So how do we do that? How do we found the bioactive compound derived from a mushroom for the cancer? Maybe finding a curing for a cancer, Professor.
Yeah. By using the high screening process.
Oh.
Yes, all extracts try to do the screening.
Okay.
Yes, okay.
So the answer is through a screening,
Yes.
and then after the screening?
Yeah. Then select the extract and the isolation.
Oh, after the screening, we extract and then we found it.
Extract numbers, so then purified analysis.
Oh, okay. And then continue biopurified, and then after that, we got those chemical compounds that maybe with another analytical process, it
by
can.
using the LCMS or LCNMRs by using the yes.
Okay, by using another path, I think it can be like a promote into like a curing for cancer. I think that's all, Professor. And then I'm really sorry because our time is limited. I think I should be ended this conversation. And thank you so much. And once again, Professor, thank you so much for it today. Okay, so please everyone, I think we should give applause for Professor Ichiyanagi. Okay, so please, okay. I think before I end up my role as a moderator today, I have a pantun that I want to deliver to all of you. So for those who does not know what pantun is, Professor, so pantun is basically like Indonesian poetic. Okay, it's usually like delivering in 4 lines in total. Okay, so but I think I should need a help from all of you. Instead of saying cakep, because we still have a Japan professor right now with us, is it I ask what is cakep means, and the answers is from is a kire. Am I right? Yeah, kire. So please do not say cakep, but instead of cakep, every time I finish with 1 line, help me with say kire. Is that kire means beautiful, Professor? Beauty? Kire means beauty? Beauty, right? Okay, so I'm gonna have a pantun. Is more like a poetic Indonesian expression actually. Okay, maybe it will be delivered in Bahasa Indonesia because it's a part of the poem in here. But instead of saying cakep at the end, we're gonna respond it with say kire. Okay, kire. Okay, everyone's ready? Okay, so allow me to switch on to language for a moment. Okay, so mencari jamur ke dalam hutan. Kire, not cakep. Okay, let me repeat. Someone has just wrong in answering me. Okay, so mencari jamur ke dalam hutan. Okay, thank you. Jamur ditemukan setelah hujan. Selesai sudah rangkaian kegiatan. Sampai jumpa lagi di lain kesempatan. Thank you so much everyone, and assalamualaikum warahmatullahi wabarakatuh.
Okay, let's give another round of applause with the beautiful poetry or pantun that coming from Miss Hikmah, and also thank you Professor Ichiyanagi for presenting another knowledge and insight to us all here. And to all participants here, the next session, as a token of appreciation to our speakers and moderator who already supporting this visiting professor event, we would like to invite to both of our speaker, Professor Shimomura and also Professor Ichiyanagi, to come to the front, stand on the center of this kind of little stage of us, because there's a little present for Faculty of Science and Technology of Universitas Terbuka. You can stand in this side and facing all of the audience here. And I would like to invite with honor our Dean of Faculty of Science and Technology of Universitas Terbuka to deliver a certificate of appreciation to both of our speaker. Okay, so Dr. Nurma, this is the Dean of Faculty of Science and Technology at Universitas Terbuka, will deliver the certificate of appreciation. First is for Professor Norihiro Shimomura. Okay, and the next certificate of appreciation will be delivered to Professor Tsuyoshi Ichiyanagi. Okay, please remain on stage because we still have another present that coming from Universitas Terbuka. This is the iconic merchandise that coming from Universitas Terbuka, and also the traditional, what do we call it? Traditional things coming from Java, another island in Indonesia. We call batik. So, and kain tenun. This is a little present coming from Indonesia and also Universitas Terbuka that we will present it to our both speaker that already giving us such an insightful knowledges and informations and also knowledges to us to all here. Okay. Okay. Wow, already using the UT cap here. It's really good and sits on you. Thank you, Professor Shimomura. If you may, Professor Ichiyanagi, you can use the cap from Universitas Terbuka. Okay. And last but not least, a little plaque that coming from Universitas Terbuka that will be maybe it can be presented at Tottori University, a little plaque for both of Professor to Professor Ichiyanagi and also Professor Shimomura. You can present it at Tottori University that you already coming to Universitas Terbuka. Okay, and another one, a little appreciation to our moderator, Dr. Iwan Saskiawan. If you may to come to the front, we do have a little present to Dr. Iwan Saskiawan who already guiding us through an insightful session today with Professor Shimomura. Okay. A little iconic merchandise that coming from Universitas Terbuka. If you may, okay. So we still gotta have another photo session. So I would like to invite Dr. Rini, all of the Vice Dean at the Faculty of Science and Technology to come to the front to have another photo session. Because the first photo session, we're not having a photo session with our dean. So in this session, we will have another photo session with our lovely Dean of Faculty of Science and Technology. And also I would like to invite the head of Biology Study Program and another moderator, our birthday girl today, Mrs. Hikmah. Okay. Okay. So same with our previous photo session, we will have a formal, a semangat pose, and also UT pose. Okay, so we will start the formal pose. Oh, okay, Dr. Pepi. Okay. 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 If you want to move a little bit to the left side. The left side, okay, step to the left. Okay, sudah okay? Okay. Okay, the first session, we will count down from 3, 2, 1. Okay, the next pose is semangat pose. If it's possible for you, Professor, it's okay, okay? Okay, we will count them from 3, 2, 1. And the last one is salam ute. Is that okay? Okay. Oh, the cutest pose of Salam UT. Okay, thank you, professors. Okay, we will count down from 3, 2, 1. Okay, let's give another round of applause. Thank you, sir, madam. You may have your seat again. So, ladies and gentlemen, we have reached the end of today's visiting professor event. And on behalf of the committee and the whole faculty, especially the biology study program, we would like to extend our gratitude for our speakers, Professor Norihiro Shimomura. Arigatou gozaimasu. And next, Professor Tsuyoshi Ichiyanagi. Arigatou gozaimasu. And our moderator, Dr. Iwan Saskiawan and Ms. Hikmah Zikriani. Once again, thank you for guiding us such an insightful and more knowledges that we had today. And all of the faculty officials, committee, and all of the participants for your contributions on this event. And I would like to inform to all participants, don't forget to fill the attendance form by clicking the link that already shared by the committee at the chat box at the Zoom. And for those who attending this event physically here, you can scan the QR code that already prepared by the committee at the registration table. Jadi untuk hadirin yang hadir secara luring bisa mengisi melalui scan QR code yang sudah ditampilkan oleh panitia ataupun sudah disediakan di meja registrasi. Dan untuk partisipan yang mengikuti secara daring bisa langsung klik link yang sudah dibagikan oleh panitia. So after filling the attendance form that already prepared by the committee, all of the participants will get certificate of appreciation. So don't forget to fill this attendance form. And then in a few minutes or in a few days, you will receive the certificate of appreciation for this visiting professor event. So we all hoping that today's event became a great experience for us all here to share knowledges, experiences, and also inspirations. So once again, my name is Safira. Thank you. Have a good day. We're signing out. Hope to see you again on the next visiting professor. Thank you.
Recording stopped.